Welcome to today's Explainer. We're diving straight into the fascinating structural world of lipids, which are honestly nature's most versatile building materials. If you've ever wondered how your cells actually build their protective walls, how your body manages to store its deepest energy reserves, or even just why oil and water absolutely refuse to mix, well, you're in the right place. Today, we're going to look at the exact molecular blueprints that make all of that possible. Here's our roadmap for the tour. First, we'll meet the lipid family as a whole. Then, check out steroids and phospholipids. After that, we'll hit triglycerides, break down their structural building blocks, compare saturated and unsaturated fats, and finally, look at how cells assemble and tear down these molecules. All right, Section 1. Meet the lipid family. Let's start by figuring out what we're actually looking at here. The really crucial thing to understand right off the bat is that a lipid isn't just one specific shape or structure. It's actually a super broad category of biological molecules. We're talking steroids, phospholipids, triglycerides, and they are all grouped together for one main reason: they are predominantly hydrophobic. In plain English, they are absolutely terrified of water. They just cannot mix with it, mostly because of their non-polar carbon-hydrogen bonds. And honestly, that one single chemical quirk is the foundation for literally everything we're about to talk about. Moving on to Section 2, steroids and phospholipids. Let's step into our first couple of structural neighborhoods. Let's talk steroids first. When you think of cholesterol nestled right inside a cell membrane, you're looking at a classic steroid. It's an absolutely essential component that helps regulate how fluid and flexible your cell membranes are. But it doesn't stop there. Cholesterol also acts as the baseline precursor for synthesizing steroid hormones, things like estrogen and testosterone, which go on to coordinate basically your entire body's daily activities. It's a massive multitasker. And then we have the phospholipids. These are the molecules that form that famous bilayer, acting as the primary architectural boundary for every single cell in your body. Because of their incredibly unique shape, they carefully wall off the delicate, complex machinery inside the cell from the completely unpredictable environment outside. You can think of them as the ultimate biological security guards. Section 3 brings us to triglycerides, nature's deep energy reserves. This neighborhood is probably going to feel a lot more familiar. This is literally where your kitchen connects directly to your cellular biology. Triglycerides basically come in two main flavors, depending on how they act at room temperature. You've got your solid fats like a stick of butter, and your liquid oils like olive oil. Now, even though they look and feel incredibly different to us, structurally, down at the molecular level, they belong to the exact same family. And their job goes way, way beyond just making food taste good. Triglycerides are your body's alternative energy vault. In fact, a single gram of fat stores significantly more energy than a gram of carbohydrates, all thanks to those dense carbon-hydrogen bonds. Plus, these same molecules provide thick thermal insulation. Think about marine mammals like whales surviving in freezing oceans. They also act like biological shock absorbers to protect your internal organs, and they even give aquatic life natural buoyancy. Pretty wild, right? Let's hit Section 4, the building blocks. To figure out why fats pull off all these tricks, we have to look under the hood. You're probably wondering at this point, what exactly are the microscopic puzzle pieces that snap together to make something as complex as a triglyceride? Well, it boils down to two main components. First up, glycerol. Glycerol is an alcohol with a neat little 3-carbon backbone. And attached to each of those carbons is a highly reactive hydroxyl group that's an oxygen bonded to a hydrogen. You can basically picture glycerol as the central anchor, or the scaffolding, for the whole fat molecule. Bolted onto that glycerol anchor are the fatty acids. A fatty acid is basically a long, unbranched hydrocarbon chain, literally just a long string of carbons and hydrogens, and it ends in a very specific carboxyl group. Now, here's the absolute game changer. This specific physical structure gives fatty acids a total split personality. The scientific term for this is amphiphatic. That carboxyl head at the end, it loves water. It's hydrophilic. But that long hydrocarbon tail, terrified of water, extremely hydrophobic. The sheer length of that nonpolar tail is exactly why fats and water simply will not mix. The tail wins the tug-of-war. Which brings us perfectly into Section 5, saturated versus unsaturated fats. That long tail we just talked about holds the whole secret here. When you compare the two, it all comes down to the geometry of the carbon bonds in that tail. Saturated fats only have single bonds between their carbon atoms. That means they are fully saturated with the absolute maximum number of hydrogen atoms possible. They're straight, so they pack tightly together, making them solid at room temperature, like animal fats. Unsaturated fats, on the other hand, have at least one double bond in the chain, so they don't have the maximum number of hydrogens. That double bond creates a literal kink in the molecular chain. They can't pack tightly, so they stay liquid, like your plant and fish oils. All right, final stop, Section 6, Assembly and Breakdown. Let's watch the actual cellular construction crew at work. Building a triglyceride is a highly sequential, incredibly mechanical process known as a condensation reaction, or esterification. Step 1. The cell lines up one glycerol with 3 individual fatty acids. Step 2. A water molecule is physically squeezed out and removed. Step 3. Because that water left, a super strong bond called an ester linkage forms right between the glycerol and the fatty acid. But the cell doesn't just do this once. It repeats this exact condensation reaction 3 separate times. The first connection gives you a monoglyceride. The second one makes a diglyceride. And that third and final reaction yields your full triglyceride. The whole process sheds 3 water molecules and leaves everything locked tightly together by 3 brand new ester linkages. But what goes up must come down. When your body needs to actually use that stored energy, it runs the demolition in exact reverse. It uses a vital biochemical process called hydrolysis, hydro meaning water, and lysis meaning to break or shatter. By forcefully ramming 3 molecules of water right back into the structure, those strong ester bonds are completely shattered. The triglyceride gets blown apart, cleanly separating the glycerol from its 3 fatty acid tails. And now, those tails are totally free to be metabolized by your body to keep you moving. So as we wrap up this explainer, I want to leave you with kind of a profound thought. Right now, literally as you sit here listening to this, millions of these complex condensation and hydrolysis reactions are happening inside you. How does this invisible, relentless cycle of construction and demolition perfectly balance itself to sustain your life every single second of the day? It's definitely something to chew on the next time you pour a splash of olive oil. Thanks for joining me on the tour today, and I'll catch you in the next explainer.